Good morning, good afternoon, good evening. Thank you very much for having me here. I'm very honored to talk today shortly about frontiers in immunotherapy, specifically CAR T cells, why they are translational frontier. I am Ivana Radosavljevic, I have a master's in cardiovascular science, and I'm currently working in immunotherapy company with CAR T cells on a daily basis with commercial and clinical CAR T cells. I'm also a member of student organization Nucleate that helps academics become biotech leaders and build their own businesses. So why do CAR T cells still matter? They are a benchmark platform for engineered cell therapy. They have a major impact in B-cell malignancies. There is a proven clinical activity and durable responses in selected settings, and still evolving biologically and operationally, because research is done every single day and the new things are discovered preclinically. This machine I'm showing you here is Clinimax Prodigy by Miltenyi Biotech. This is amazing, powerful machine for cell separation that helps separation of PBM, of leukapheresis, from leukapheresis T cells, CD4, CD8, and other cell types that can be simultaneously, but only one batch. So for one patient at a time can be done. But all steps can be done in one machine, and it's fully automated, and it's not fully automated, but it's an enclosed system. It's semi-automated, and it's that means cell isolation, separation, activation, expansion, also transduction with the virus, and then it shortens vein-to-vein time. If I show you here why CAR T cells and then all cell and gene therapy that are approved by FDA, we can see that regarding counts. So quantities, actually CAR T is the least because it's only 7 FDA approved, while for other like cord blood cells, gene therapy viral vectors, and other cellular tissues are more, for instance, close to 17. TILs here belong, for instance. But CAR T cells are still the frontier because it's not all about numbers, because so much research is going on. There's so many clinical trials currently, and they are so expensive to produce, manufacturing, and they have so much potential. They're expanding in so many different fields. Where the field stands. CAR T cells are revolutionizing hematologic malignancies, myeloma, and lymphoma. There are 7 FDA-approved therapies. More than 90% is response rate in refractory relapsed B-cell lymphoma. In 2017, there was the first approval of disaglenslew-cel. For solid tumor cell, it is still a major unmet need, and translation of CAR T cell therapies is constrained by biology, manufacturing, and delivery. Now we go to CAR T in solid tumors. It has strong preclinical momentum, 41 clinical trials. Limited clinical success so far. Why? There are several barriers. Antigen scarcity. Antigens are not abundant and broad like in hematological malignancies. They are also intracellular, while CAR T cells can only target if it's superficial. Immunosuppressive TME, tumor microenvironment, including physical barrier, hypoxia, nutrient deprivation, and changed metabolism, the presence of myeloid-derived cells, presence of immune suppressor cells like Tregs, and tumor heterogeneity. In one tumor, solid tumor, not every cell expresses same antigen and does not express same antigen in the same quantity. And also among different patients, so for the same, let's say, type of tumor has actually way different tumor heterogeneity, antigen expression. Then there's off-tumor toxicity. Most known case is HER2, a clinical trial when they gave to a patient, but it ended up for some other, I think it was for, I'm not sure, was it a breast cancer, and it ended up in lungs because HER2 is expressed in lungs. Lungs were in that case healthy tissue, and the patient sadly died. Here is circle that is showing us different targets for antigens for solid cancer. So for instance, for colorectal, we have CEACAM5, HER2, MAC1, but then again, we see that antigens are repetitive, like MAC1 is expressed in colorectal and also in ovarian and cervical cancer. HER2 is expressed in colorectal and in liver. And also MAC1 is in liver. And then pancreatic cancer. Then mesothelin that is expressed across the lung, mesothelia, ovarian, cervical cancer, pancreatic cancer. That's not good for targeting, as you may notice. And as already mentioned, the tumor microenvironment is suppressing. So we have a suppression through Tregs, through myeloid-derived cells, PD-L1, then metabolic and physical constraints with hypoxia. Oxygen is deprived where there is a cancer. And also there is a nutrient deprivation that is affecting T cells. T cells need energy to be active. This is why we have some innovative strategies, like for instance, hypoxia-driven CAR T cells. So CAR T versus TCR-T. TCR-T is particularly promising in solid tumors because it expands the accessible antigen repertoire. Targets intracellular antigens via HLA presentation, while CAR-T cannot target intracellular. CAR-T is superior for direct surface antigen targeting, and those two are complementary platforms. I show you in this table. Because targets for CAR-T surface antigens only, for TCR is intracellular antigens, neoantigens. MHC dependence for CAR-T, luckily, they are MHC independent, while TCR-T are very MHC dependent. Potency, CAR-T are strong, potent short-term killing and high cytokines. TCR-T are better expanded under high antigen load. The good part is there's less exhaustion, but it requires high antigen load that can be problematic because for CAR-T, high antigen load is not needed. And high antigen load means that antigen has to be abundantly expressed, and for many solid cancers, that's not the case. Solid tumors for CAR-T is limited, but for TCR-T is more promising. There are clinical trials. Risks for CAR-T is higher CRS neurotoxicity. For TCR-T is similar, but potentially milder. And approvals, sorry, this is a mistake. 7 CAR-T are approved for cancer, and TCR-T is not yet approved. It's still an R&D pipeline. We go to allogeneic CAR-T. So all FDA approved are autologous, but we have allogeneic. That means they are produced from another patient. The aim of having allogeneic CAR-T is off-the-shelf availability. And advantage is speed, scalability, and standardization. What is happening here? From a healthy donor, T cells are taken. They are genetically engineered in the lab or in the clean room, and then they are transferred to a patient, to a cancer patient who has cancer, so that those CAR T cells can target cancer. Main barrier with having allogeneic CAR T is graft versus host disease and host rejection, and persistence remains a critical challenge. So what are some of the strategies for allogeneic systems? I believe this slide shows really beautifully. First, one of the ideas TCR knockout to reduce graft versus host disease, then HLA engineering to limit host rejection, then alternative immune cell sources, like here is showing NK are taken, so it doesn't have always to be T cells from PBMCs. NKs, which are then viral transduced, and then there is CD19 CAR expression, perhaps also interleukin-15, because interleukin-15 is for activation and rejuvenation, and then also caspase-9. Why is caspase-9 here overexpression? This is basically for switchable. This is, I believe, for modular, not CAR-T, but modular CAR-NK therapy, because it has switch on and switch off. So in this case, interleukin-15 is switch on, and caspase-9 is switch off. So once CAR-NK is done with kill the cancer with its activity, it can switch off the caspase, overexpressing the caspase, then destroys activates the NK cell. And also there's gene editing as an enabling technology. We also see iPSCs. I'll talk about them on the next slide, which are very powerful. And NKT, so we have, for instance, allogeneic CAR therapy, allogeneic NK CAR, and allogeneic NKT CAR. So NKT are natural killer T cell. This is a special cell type that's sitting in between innate immune system and adaptive system. And they're really powerful. iPSC-derived CAR-T, they're very powerful. Start with totipotent embryonic stem cells, then you go to pluripotent, multipotent. Basically, can be different depending on which stage we take them. They can be differentiated into virtually any cell type. So the renewable and scalable cell source, clonal standardization is possible, potential for industrial manufacturing, and still early in translation and not yet routine. Here, I want to stress ATPSC, adult telomerase positive stem cells, is a cell type that has not recently been discovered, but still not that popular, didn't become a routine. So I talked to the founder, Henry Young, who basically patented them, who published so many papers and protocols on isolation, and it has a broad application, this cell type. The main problem with ATPSC is that they are in such a small amounts present in a human body, but they are very powerful. They can be reprogrammed into CAR T cell. That's one of the ideas. I actually talked to him, and he's not CAR T expert, so he doesn't know, but he believes it's possible. And the thing is, it's not patented. We even don't know if there is research going on. At least there are no publications. Nobody started. We don't know why. I'm also not expert in stem cells, so I cannot speak why is that happening. I don't know. In vivo CAR engineering. What is the idea with in vivo? To generate CAR T cells directly inside the patient. So, how does it conventionally go? We have ex vivo autologous and ex vivo allogeneic types. So in autologous, first leukapheresis blood is taken, then T cells are isolated, then they are with the viral construct transduced, with the chimeric antigen, the viral helps the transduction, then we have CAR T cell, which is injected back to the patient. In the allogeneic donor, healthy cells are taken, then again lentivirus approaches and CRISPR engineering to get the CAR T cell, and then it's injected in a patient. The main idea is to speed up this process. So to bypass ex vivo manufacturing, which can be potentially transformative for accessibility. What they are doing is they're using mRNA encapsulated in LNPs. Or also lentiviral delivery system doesn't have to be LNP mRNA. It could be mRNA, CAR-RNA genome in a lentivirus, for instance, or even AAV-based delivery. Or this biodestructive scaffold that it's injected in the human body and then it activates, so it integrates itself in a CD8 T cells in the human body. So safety, specificity, delivery still remain unresolved. I have to shed the light on this recent paper in Nature Medicine of in vivo generation anti-BCMA CAR T cells in relapsed of refractory multiple myeloma phase 1 study in China. And I read the abstract because I don't have access to the whole paper. And then I go to the clinical trials gov.net website to read about the clinical trial, and I see that it's halted. They claim this is successful. It's successful in terms that we have finally proved that in vivo CAR is possible. It happened in a small group of patients, people in this whole world. And the problem is that they all developed third grade of side effects, and they also developed cytokine release syndrome. One patient died from neurotoxicity. So, and the trial is halted. It means that it's still not safe to be administered. So it's effective, but it's not safe. So of course, that's why we have next stages of a clinical trial to test this safety and therapeutic window. Why is translation so slow with CAR T therapies? When CAR T therapies manufacturing per se is a bottleneck. It's complex. There are cost and logistics. So first, when it comes to this isolation, so investors are worried since it's okay. There was that amazing machine learning model that I'm showing here, but it's only one batch, and then that's costly and that takes time. That means that you have to have machines as many patients as you have. If you have to have 20 productions per day, you have to have a clean room with 20 clinimax, and that means 40 people because the minimum is 2. It's always more, but the minimum is 2 people per workstation because of COI check. And then there is a risk of mixing CD4 and CD10. Later, also with the virus, there is a risk with also the contamination and the safety risks. The cost and logistic, that's all very costly. And since it's autologous production, there is a risk also of cryopreservation. It must be cryopreserved. The cryopreservation affects the quality of the product. Basically, these cells lose viability. CAR T cells lose viability with the cryopreservation. and then that vein to vein time is really long. Exhaustion is senescence. The biggest problem here is the many time starting material. We even don't know. We don't have special tests. We do not screen patient when a patient is scheduled. The blood is already drawn. Leukapheresis arrives to us. That's how it works. There is no, because doctors, clinicians have other criteria how they exclude patients, but they don't know biologically speaking what is the viability, what is the exhaustion and senescence state. And the problem when cells are exhausted in senescent, they're not going to be effective. They are going to be CAR T cells that express chimeric antigen on flow cytometry. There'll be a proof, yeah, it's your blood, but they're not going to kill the cancer. Metabolic dysfunction and loss of fitness, I'm going to speak about metabolism a bit more, and slow movement from clinical to clinical research. What's the worst here? I say that actually it seems that academic research is faster. They always say industry is faster. It's fast in following trends because commercial, it follows money, and academia is slow, it's standardized, it never changes. However, the preclinical research is much faster than translation is super slow because of so many regulatory agencies, and the whole idea of GMP just means it takes so much time to convert something experimented there into GMP. So this is why we have allogeneic therapies, some of alternatives and lab automation that can help speed up the process, but sadly, until we reach the point when we don't need operators in clean room, when production will be clean room free for people, so hands free, that is not science fiction, but probably it's going to take a decade. Metabolic control of CAR T function. Metabolism shapes proliferation, persistence, and effector function. And this illustration beautifully shows this. So we have different states of T cell, naive T cell, effector T cell, and memory T cell. And when it comes to mitochondria, the powerhouse of a cell, in metabolism, in an naive T cell, it's all balanced. The same amount of glycolysis, of lipogenesis, fatty acid oxidation, and of glutaminolysis. However, naive T cells not what we care for because they're naive. They have not been exposed to antigens. They are not killers. They're not effective effectors. They do not secrete cytokines. Then we come to effector T cells, which are known to have strong glucose and glutaminolysis, but they do not have fatty acid oxidation. So really how it progresses is from strong glycolysis toward little oxphos, but ideally what we would want is to have more oxphos and less glycolysis. And memory T cell has very strong fatty acid oxidation, stronger than glycolysis and glutaminolysis. So memory-like states correlate with better durability. Glycolysis, oxphos, mitochondrial integrity are key variables, and exhaustion is partly a metabolic failure state. Recent metabolic findings. Mitochondrial dysfunction impairs ROS homeostasis and survival. Tumor microenvironment suppresses metabolic flexibility, solid tumors impose system metabolic field effects, and exhaustion is linked to poor energy production and persistence. And this illustration shows us really beautifully different states. So of course that on the picture A, CD4 and CD8 ratios are shown because CAR T cells are formulated that they're given in 1-to-1 ratio as a CD4 and CD8. T cell differentiation, as I said, it starts with T and naive T cell, central memory in the effector. And there are different markers. Of course, there is a shift in metabolic shift and also shift in markers that are expressed. So we have two different states, exhausted and senescent cells, and I would love to stress, I have to explain this because people get confused always. Even I used to be confused at the beginning. It's really important that I show here that we're speaking of two different things. T cell exhaustion it's not good per se, but it's easier to do damage control than with T cell senescence because this is reversible, this is temporary. Cells can be exhausted and then reverse to normal effector state. So exhaustion means expressed TIGIT, PD1, LAG3 and TIM3, there is high glycolysis, there is low OXPHOS. What does it mean if a cell is exhausted? It's loss of effector function, loss of cytokine production. It's temporary. Then we have senescence. As the word says, so it's getting old. It's the same like with our aging, with the humans. This is irreversible. This is a permanent state. So when doing flow cytometry, they're showing viability in counting T cells. Those CAR T cells in the blood, patient has them. Tests show there's a lot of CAR T cell. They're in correct effector target killing rate. There's no cytotoxicity. Why? Because they're already senescent. They have high express CD37, KRG3, CD27 and CD28 are decreased. CD28 is co-stimulatory domain that is responsible for fast and strong activation, while CD27 is also used as the co-activatory molecule. So it's CD3 and then CD27 in laboratory when CAR T cells are isolated. First, in order to be active, they activate with CD3, CD27 bits. And senescence means that the transduction efficiency, expansion, and cytotoxicity, all of those are reduced permanently. Translational metabolic engineering. Metformin, interleukin-15 programming, enhanced metabolic fitness. As we already mentioned, interleukin-15, that it could be rejuvenation, but it's not against senescence, it's actually for metabolic engineering. So also there is mTOR pathway, which tuning preserves memory phenotypes, and metabolic reprogramming is becoming now design principle for CAR T cells, which is really amazing, because what I want personally to stress, and it's not enough emphasis given, is that this is a whole like architecture and the framework. If we only isolate one component and then change it, modulate it in the lab, like for instance engineering of the CAR T itself, just designing tandem receptors, or modular strategies, while not focusing on what really matters, like metabolism, then we're gonna not have very successful therapy because important link is missing. So in conventional CAR T cells, as already mentioned, we have 3 different pathways: glycolysis, mitochondrial biogenesis, OXPHOS, and fatty acid oxidation. With this new therapy, with metformin treated, so they are treated CAR T cells and then they're infused in a patient. Metformin is a metabolic regulator that activates STAT3 pathway, which is great because it upregulates fatty acid oxidation, and also through ERK1 and 2 signaling, which upregulates mitochondrial biogenesis, OXPHOS. That means less differentiation, increased activity. It blocks STAT5, and STAT5, the signaling transduction, leads to glycolysis. But if STAT5 is blocked, inhibited, then glycolysis decreases naturally. And as we switch from glycolysis to OXPHOS, we have increased anti-tumor activity. That's why it's so important. Modular and logic-gated CAR T. This is now my favorite part. Traditional CAR T cells are single antigen, means it's always on. So the activation is on. Once it's infused in human body, it's going to be active and there is killing happening. Modular CAR T are 2-component system. Now we have on and off. So we have universal CAR T cells, and another part is that adapter, which is single chain fragment region switchable, which is actually plus a bit of hinge, is sorry, not a hinge, it's a switchable module with a switch molecule. I meant that a switch molecule binds to the hinge on a CAR T cell, uniCAR. Conventional, yeah, excuse me, it's all together. And what we also have logic gating. Here applies Boolean logic, and or not, to CAR input signals. What does it mean if it's AND signal? I'm not sure if you can see because my face is here. Is that both A and B antigens have to be here. If they are not, then CAR T cannot bind to the cell. Or gate means that one of those can be active, A or B, then it binds. Not gate means that if one is present, then it cannot bind. For instance, B then it blocks the binding. If it's not present, but if it's not present, then the binding happens. What will define the next decade? So, as already mentioned, multi-targeted logic-gated CAR-Ts, armored microenvironment resistant CAR-Ts. So when it comes to armored, that's, for example, they are engineered to produce cytokines that will block PD-1 inhibition, or because PD-1, PD-L1 is a checkpoint inhibitor that actually deactivates our immune cells, CAR-T cells, so it has to be blocked. Or metabolic reprogramming and memory state preservation. And the allogeneic, as already explained, allogeneic iPSC-based manufacturing, maybe in the future we will see ATPSC based CAR-T cells and in vivo engineering as a disruptive path. And here this illustration again shows logic gating, for instance, and then tandem CARs and different gates that protect, like not gate, for instance, protects the healthy tissue. So thank you so much for your attention. CAR-T has already changed immunotherapy field, but the real frontier is how to extend, simplify, and industrialize this platform. And this is exactly where the next decade of progress will be made. And of course, clinicians here are really important because they are the people who are in direct contact with patients. So thank you so much for your attention. I hope that I will later be given questions.